So this script is a script that you can plug in onto the basic script. The goal of this script is to check if there are any cell cycle effects in the data. So what are cell cycle effects? Well, a good explanation of cell cycle can be found on Wikipedia. So as you can see, cells, they go through a circular process that's called cell cycle, and in that cell cycle you have different phases. You have the G1 phase, you have the S phase, and you have the G2 and M phase of the cell cycle. And when cells are in different phases of the cell cycle, they will express a different set of genes. So gene expression depends on the cell cycle phase a cell is in. So that means that when you have in your sample cells that are in different phases of the cell cycle, that you will have differences in gene expression between these cells. But these differences in gene expression do not mean that these cells are different cell types. No, it simply means that they are in a different phase of the cell cycle. So these variations in expression that are caused by the cell cycle, they can obscure other possibly much more interesting sources of variation in gene expression. So because of this, you might miss differences between cells that are biologically much more interesting than the cell cycle itself. So you want to get rid of this variation that is caused by the cell cycle. And that's exactly what we're going to do in this script. How are we going to do this? Well, for each phase in the cell cycle, we have a set of marker genes. So a set of genes that are only expressed in that specific phase of the cell cycle. So you have marker genes for the S phase, and you have marker genes for the G2 and M phase of the cell cycle. So in each cell in our data, we're going to check if these marker genes are being expressed, yes or no. So if a cell expresses marker genes for the S phase, it's considered to be in the S phase. If a cell expresses marker genes for the G2 and M phase, it's considered to be in the G2-M phase. If a cell doesn't express any of the known marker genes, then it's considered to be in the G1 phase of the cell cycle. So, and this approach was first described in this paper. I'm not going to go into detail here, but you can take a look yourself.